So I am going to invite Dr. Burdeja, who is going to talk about what has certainly become the hottest topic in not just myeloma, but in a lot of areas. Dr. Burdeja comes to us from the Sarah Cannon Research Institute in Tennessee, and I'd like to say that he is one of the best kept secrets in the myeloma world. This is actually one of the people who have made more new therapies available to myeloma patients than anybody in the country, and this particular topic will be transformative. So thank you, Lindsay.
Thank you so much. Thank you so much for having me. It's nice to leave my 75-degree weather to come to your 50-degree overcast weather. But aside from that, it's a pleasure to be here. And it's actually interesting that I get to give the only talk that's going to have no therapies that are available to you as standard of care. So just to echo David's points, is the importance of clinical trials is not only to sort of help us move the field forward, but it also really helps you to have availability for therapies that are on the verge of hopefully becoming therapies that can be made available to all. The sooner all of you join clinical trials, the sooner they become available to all of us. So again, a plug for clinical trials. So my topic is on immunotherapies, not just CAR T therapies. So we'll talk a little bit about just the immune system to begin with, but I just put a list up there of the different immune therapies that are available. And David already talked to you about the monoclonal antibodies, daratumumab and elotuzumab, so I won't actually go into those. But there are other antibodies in development. But we'll also start talking about just not the antibodies, but antibody-drug conjugates, perhaps bispecific antibodies, antibodies that may have multiple targets. Of course, the cellular therapies, CAR Ts being one of them, vaccines as well. I am not going to talk about the immune checkpoint inhibitors, although you'll hear a lot about that. There was actually a question in a previous I saw that was submitted by someone that when a patient is in a checkpoint inhibitor, these are therapies that are still being worked on, but there's been a little bit of a snafu in myeloma, and it's unclear where we're headed with those. So, but I'm happy to answer questions later on if somebody has them. So the immune system is incredibly complicated, and it's a good thing because that's your defense towards pretty much everything. And so we think of the immune system when we get an infection, right? That's what's helping you fight infections, and so it's defending you from bacteria and viruses and fungi. But it's also what's actually defending you from getting cancer. And so I think we don't think of it in that sense. The immune system is supposed to actually get rid of anything that doesn't belong in your body. But for some reason, sometimes it goes dormant, and it allows things to develop. So when we talk about the immune system, we talk about the innate immune system, and it's sort of what you've heard a little bit earlier with the antibodies. So these are sort of like the prime defense. These are, you have antibodies that defend you against incoming bacteria, for example, that act very quickly. But then you also have the secondary immune system that actually learns from that initial invasion to try and keep you from getting the same process again. So it kind of can build a more robust response as well as actually lay dormant and then become active when needed. So how come our immune system is not getting rid of our myeloma cells? Well, one of the problems is that myeloma cells are actually born out of normal cells. And so for some reason, the immune system doesn't see them as foreign. The cells look too much like ourself, which the immune system has been taught not to go against. The other thing is myeloma cells actually can make these proteins that can actually help it hide from the immune system. They basically form these barriers where the immune system is told, no, this is a cell you do not want to target. Myeloma cells can recruit parts of the immune system that normally would lead to tolerance of the immune system. And so there's all these changes, all these cellular changes that are going on in the milieu of your bone marrow where your myeloma is found, all these different things that are basically protecting the myeloma cell. And so for many reasons, it is very difficult for the immune system to get rid of myeloma on its own. So in terms of how do we target, then how do we help your immune system fight your myeloma? There's different ways that you can do this. And so you've heard about the monoclonal antibodies, which are here somewhere. There. No, never mind. So you heard about the monoclonal antibodies. And as David suggested, these are antibodies not too dissimilar from the antibodies your myeloma cells make, but these are antibodies that presumably are going after a particular target, in this case, your myeloma. So those are called monoclonal antibodies, and we'll talk a little bit about those. You also have the IMiDs or the checkpoint inhibitors, which we're not going to talk too much about, but these are basically drugs that are trying to decrease the amount of proteins that the myeloma cells are making to help defend themselves against the immune system, or try to unmask the myeloma cells so that now they become visible to your immune system. And so that's sort of the idea behind those. You can have vaccines, no different from when you get a flu vaccine. So a vaccine presumably should be able to elicit an immune response, telling your immune system to go after your myeloma cell. And so that's the idea behind the vaccines. But vaccines are still dependent on your immune system being active. Or you can go to something which is more like a T-cell type therapies, like CAR T therapies, where you say, your immune system is not working well enough, let's make it work enough. And so let's change it enough to go after your myeloma cells. And so that's a little bit of the idea behind CAR T cells. So in terms of the antibodies, you heard about the naked antibodies. That's your daratumumab and ilotuzumab. These are antibodies that are targeted to go directly after a particular protein. So in daratumumab's case, it goes after CD38, which is expressed in myeloma cells. The drug conjugates, or the antibody drug conjugates, is an antibody very similar, but it's also attached to a toxin or a radioactive or radiation. And so the idea here is that it's almost like a Trojan horse, where you have the antibody bind to the myeloma cell ingests the whole complex, and the chemotherapy that's attached to it is then released inside the cell. And so it's more of a targeted type of way to deliver chemotherapy or radiation. And then we also have these things called bispecific antibodies, which actually are almost like, think of them as two-headed antibodies. So one of the heads goes and attaches to a myeloma cell, the other head goes and attaches to your T cells, it activates the T cells, it brings it into close proximity to the myeloma cell, and it basically forces the T cells to kill the myeloma cell. So that's the idea behind the bites or the bispecifics. In terms of monoclonal antibodies, again, I'm not going to go through daratumumab or ilotuzumab, but there is another anti-CD38 monoclonal antibody called isatuximab that is also in clinical trials. It has very similar activity to daratumumab. It is currently undergoing phase 3 trials in combination, and it's looking very promising. So likely a drug that also will make its way into the market not too far from the future. And at this last ASH that we had, which is our national meeting in the United States, in December, Dr. Trudell presented very provocative data on this new antibody drug conjugate. And again, this is an antibody that is bound to a toxin. So the antibody is directed towards this protein called BCMA. Now, BCMA is going to be a very important protein, so remember that, because all of the CAR T therapies right now are going towards BCMA. I was going to say all, but most of the ones in active clinical trials are directed to BCMA. So BCMA is this protein that is found on the surface of myeloma cells, and it is thought to be present in all myeloma cells because it is a protein that is required for the myeloma cell to become a myeloma cell. So it is only found in plasma cells. I'm sorry, I should say myeloma cells. It's required to become a plasma cell. So it's only found in plasma cells and in mature B cells. So a very specific target, right? You don't want to target something, a protein that is found in your heart tissue, for example. Then you give this drug and then you kill your heart as well. So it's very important that these are very targeted therapies just towards your cancer cells. So BCMA, you'll hear a lot about BCMA. So this particular antibody drug conjugate is an antibody that goes towards BCMA, and it's bound to this chemotherapy, not too dissimilar from this drug called vincristine. It's a drug that we used to use, probably the Davids know a lot more about vincristine. They used to use it a lot before transplants. It used to be called VAD. We still use it every once in a while, but very rarely. But anyway, so it's a very active drug for cancer. So, but the idea here is that instead of giving the chemotherapy in the IV and have it work and have side effects, you can give a much more potent dose of the chemotherapy with less toxicity because you can target it to go directly just to the myeloma cells. And so that's what this antibody-drug conjugate does. It basically binds BCMA, binds the plasma cell. The cell then brings in the antibody-drug conjugate, and then the chemotherapy is released inside the myeloma cells. So this is the data that's been presented. It's very early in the studies, but as you can see, there's a dose escalation. And so when you're in the phase 1 dose escalation, we have to start very low because we don't know what kind of toxicities we see, and then you move up in the dose. So in the initial doses, we didn't see too much response. Once you get to the doses that we know are likely to be active, you can see that at these dose levels, 66.7% of the patients had a response. And these are all very heavily pretreated patients. So this is quite an impressive response. So most of the time when you're talking about a phase 1 trial, you're hoping for a response with your single drug of at least 20%. And so a 66% is a home run-like result. So very, very impressive, very intriguing. This is obviously still ongoing, but don't be surprised that these antibody-drug conjugates move forward very quickly. The bispecific antibodies are a little bit behind. So these are the antibodies, again, that have basically 2 targets. And one target is the myeloma cell, and the other target is your T cell. And so basically they bound to both. They bring the T cell in proximity to the myeloma cell and basically force it to kill the myeloma cell. At least that's the idea behind it. There's actually a drug called blinatumomab that is actually FDA approved for ALL, so a different type of cancer, with the same kind of concept. So we know there's proof of principle out there. Myeloma, we're a little bit behind. But as you can see, there's actually several trials that are ongoing with different bispecific antibodies targeting different proteins in the myeloma cell, not just BCMA, but also CD38, which is what daratumumab binds to, but also some very sort of new targets that have been found and are being tested. So this is all very early, phase 1, a few patients enrolled in the different trials, but we'll be seeing data at the next meeting most likely. Cancer vaccines. So cancer vaccines have been around for a very long time. But it's a very intriguing therapy. So basically the idea here is that you're going to inject either myeloma cells or some protein in the cells that will basically wake up your immune system and form an immune response against your myeloma cells. You give it back, and in theory, it should fight your myeloma. The difference between vaccines and how we normally think about them versus actual treatment vaccines is here. So when we talk about vaccines, we usually think in the infectious disease setting, right? So we gave the example of influenza. You get a vaccine, so you don't get influenza. So you're exposing your immune system to a protein that will help it develop an immunity so that if you do get exposed to the actual virus, it will be there ready to fight it and prevent it. So that's the idea behind these infectious disease vaccines. The problem with cancer vaccines is that your body is already exposed to the disease, right? So you already have it in you, and your immune system actually was not able to mount that response. And so it's been a little bit more difficult, but the idea is still the same, that somehow if you can figure out how to now expose your immune system to proteins that maybe it doesn't normally see that are part of your myeloma, that you can then teach it to then go after your cancer. And of course, the nice thing about vaccines is that very much like, let's say, your measles, mumps, rubella vaccine, where you get it and you might get one booster, you're protected for life, or at least for 10 years, would be that you would develop an immunity that can lay dormant at any time. So if the myeloma were to come back, it would then reactivate and go after it. So that is the promise of vaccines, although it still remains a little bit early. One of the things we do know is that most likely vaccines will work best when there's very little disease in there. You need time for that immune system to work and to really develop and to protect you. So if you have a lot of disease burden it is unlikely that giving a vaccine by itself is going to overcome all that disease or help your immune system really kill everything. So most of the trials being done with vaccines tend to be when you have the least amount of disease, which is often after a stem cell transplant. So we kind of talked a little bit about this, but I'll just emphasize the last point because it actually becomes pertinent also with the CAR Ts, that one of the things we would like is not only having a treatment that helps you right here and there, but also something that can help prevent it from coming back, right? Which is ultimately what we all hope. If we can get you in remission and then give you something that can prevent it, ultimately that hopefully would be the potential that you might be able to cure this. So these are just, again, I'm not going to go through these, but there's cell-based vaccines and there's non-cell-based vaccines, just different ways of basically trying to expose different proteins to your immune system. And these are just trials that have been going on with small numbers. It started since the 1990s and more recently, recent trials. The last one was just presented again at this year's ASH. And these are non-cell-based vaccines, trials that are ongoing. Again, different ways of trying to kind of create these vaccines. Actually, Dr. Nuka presented the PVX-410 vaccine at ASH, although it wasn't 2017, I think it was 2016. Yeah. So anyway, so more to come from vaccines. Again, you're giving these therapies potentially when patients are in remission, so the data takes a long time to mature. Again, going back to what David said earlier about, we don't know if someone was an MRD 10 years ago, how they're doing now because we haven't had ways to check for MRD 10 years ago. So now the reason we're all here, right, is the cellular therapies, these CAR T therapies that everyone's talking about. Well, actually, I guess we're all here to see David, but the reason I was invited was to talk about this. So, but basically, we talked about antibodies, different ways to manipulate the antibodies. We talked about basically stimulating your own immune system to go after your myeloma. Well, can we actually now take your immune system and manipulate it so it can go after your myeloma? And that's sort of what the gist is here for the immune cell therapies. So there's several different types. The CAR Ts, or the chimeric antigen receptor T cells, are the furthest along with the most data. But there's also T cell receptor engineered T cells and marrow-infiltrated lymphocytes that are also being produced and being tested in clinical trials. But we'll only talk about the first one. So what are CAR Ts? So basically what happens is if you've had a stem cell transplant, you've gone through leukapheresis where we collected your stem cells. Well, the same thing happens here, but instead of collecting your stem cells, we actually are going to collect your T cells. So that doesn't require any stimulation, which is the plus. It tends to be sort of a shorter procedures, like 2 to 3 hours. If you're on the machine, we collect your T cells. Then these T cells are taken to the laboratory, and the new DNA is infused into these T cells that forces them to now create a new receptor on their surface called the CAR, or the chimeric antigen receptor. Once these cells are created, then they're given back to the patient. And once these cells come back into the patient, then they go in and go after whatever the new receptor is telling them to go after. In the majority of the CAR T trials that you'll see, it's BCMA. So when they see a BCMA positive cell, these T cells now go after it without requiring some prompting. They start dividing and they go and kill the cell. So I'm not going to go through every trial, but I put here the 5 trials that have the most data with CAR Ts. And so all of these CAR programs are against BCMA, but they all differ in different ways. Some specifically go after perhaps 2 epitopes on the myeloma cell versus 1. Some use mouse antibodies versus others use humanized antibodies. They have different, to create a good CAR T, you have to have this these protein that basically helps the T cells divide and become much more active. And so that differs in the different CAR T programs, but ultimately they're very similar type of programs. So if we look at the data here, the NCI was sort of the first program that developed the BCMA CAR T, and they were the first to present their data. And Penn is still ongoing, as is Juno. But really, the data that really has been sort of at the forefront has been the BB2121 or Bluebird data, and then I think it was ASCO 2016 maybe. We were all sort of kind of a little blindsided by the Chinese CAR T cells, or the legend, the LCAR V38M. So I'm going to concentrate a little bit on those because I think they have the data that most of us are very impressed by. But as you can see, if you look at the numbers, they're small, so just keep that in mind. So it's still very early data. But look at the prior number of therapies. So all of these patients, and again, the LCAR is a little bit different because, and I'll tell you why in a sec, all have about 7 to 10 prior median therapies, which means they've relapsed at least 7 times with their disease. So they've gone through all the available myeloma treatments and now are coming onto this trial. In China, they don't have Darzalex, they don't have pomalidomide, they don't have certain drugs that are available to us here. And so for them, a relapse refractory patient is less heavily pretreated or has only been treated with certain classes of drugs. So just kind of keep that in mind with this data. But if you look at the overall response throughout, except for one outlier, we're talking 80 to 100% response. And in the 2 trials that, again, are the furthest along, we're talking 94 to 100% response rate. I just told you that the overall response rate of 60% in the antibody-drug conjugate was like eye-opening. We're just very impressed by it. Well, this is the reason why everyone's very impressed by this, because these are response rates that you would love to see in the frontline setting, right? You would love to see these kind of responses, which you kind of do, but again, you never talk about it in the relapse refractory setting. Look at the CR rates. So again, except for this is the very first trial. In the patients that got, and I put asterisks here, in the patients that got sufficient CAR Ts, that we think that's where there's a threshold. You need to have so many T cells infused for them to be active. So the initial patients in the first cohorts were given too few CAR Ts. And the patients that got enough CAR Ts at the active levels, these are the response rates that are being reported, and 56 to 63% complete remission rates. So quite impressive. There's a couple toxicities that are problematic. So these are not therapies, unfortunately, like everything else, without toxicity. And so 2 of the main toxicities that we worry about are this so-called cytokine release syndrome and neurotoxicity. So cytokine release syndrome, I just told you that when the CAR Ts go back into your body, some of the way what they use to actually divide and to recruit other cells and to kill myeloma cells is by releasing all these cytokines. And so these cytokines are not too different from when your body is trying to fight an infection. So if you have an infection, your immune system starts to fight it. Some people get really, really sick. They get high fevers, they drop their blood pressure, they go to the ICU. It's called sepsis. Well, cytokine release syndrome is very similar to sepsis. And so it's the same idea, right? It's killing something that shouldn't be there, in this case your myeloma, but it may overwhelm your body. And so this is a very important toxicity that can actually be life-threatening. And some patients have died from cytokine release syndrome, so we take it seriously. And as you can see, cytokine release syndrome was seen in the majority of patients. Now, the good news is that cytokine release syndrome can be as simple as just a fever. So really aggressive or dangerous cytokine release syndrome, or grade 3 or 4, is what you really want to know in terms of how dangerous are these cells. And so the good news here, at least in myeloma so far, you can see that the cytokine release syndrome grade 3 or 4 is in the 30% in the NCI and PEN trial, but in the Bluebird and Legend, we're talking about, say, 5 to 10%. So again, this is actually good. So most patients do very well. And now we know that actually there's a drug that often can reverse this process very quickly called tocilizumab that is available, and it doesn't harm your CAR T cells. So we're using it much more actively now, so we don't let people get this sick. And if you give it, you can reverse the process. And so for the most part, we're gratified by how safe, at least from that standpoint, these cells have been. But again, it's very early still to know for sure. The other toxicity that we really worry about is this thing called neurotoxicity, and that one is a little bit more difficult to explain. It's unclear why this happens, but there are patients that can get a little bit confused for like a day or two, and maybe like sundowning, you're in the hospital, and nothing else. Or some patients that actually start developing symptoms like they're having a stroke, and patients actually can have full encephalopathy and coma states where you're actually in the intensive care unit and intubated. So this is a very big potential toxicity that we worry about. The good news, again, we saw this a lot more in patients with leukemia and myeloma so far. Again, even though it says that about 20% of patients get neurotoxicity, the majority of patients get grade 1 or 2 where we don't need to do any kind of intervention. It's a very transient toxicity and it goes away. Now, but the problem is if you do get it, then it becomes a very big problem that requires severe intervention with steroids, sometimes chemotherapy, and can be a very prolonged hospitalization. So because of these two toxicities that usually tend to occur within the first 2 weeks of getting the CAR-Ts, if you're on one of these clinical trials, you get admitted to the hospital for those 2 weeks for observation. But I can tell you the majority of my patients come in and they're bracing for the worst, and they get a fever the first day and they start getting worried, and then the second day the fever comes down, and then after that they're bored to tears because now they have 10 days left in the hospital and they don't know what to do with themselves. So that's the plus. So we'll see if that remains. So where are CAR-Ts going? So obviously, there are 2 CAR-Ts that are actually FDA approved. Yeah, just 2, right? One for ALL and one for an aggressive type of lymphoma. And so everyone believes that probably the next approval may happen in myeloma. So there's a pivotal trial ongoing now by Celgene, which took Bluebird's BB2121 CAR-T trials. And so the KARMA trial is now open at the same institutions that did the original trials, including actually with Dr. Siegel. So he has this trial open. And so the idea here is that hopefully we would replicate the data we're seeing in the original trials so that it can be taken to the FDA. So, you know, there is a possibility that maybe in a couple of years we're talking about a CAR-T that is FDA approved. The Chinese CAR-Ts, I told you all the caveats of that data, different patient population, but now actually Janssen has partnered with Legend, and they're going to be bringing those CAR-Ts to the United States. And so this actually CAR-T program will be starting in the next month or 2. And so we'll see if these results that were just incredibly hard to believe are true. I remember 100% response rate, 60% remission rate. So anyway, so those are probably the furthest along in the myeloma CAR-T world, but there's several that are not far behind. So how do we improve on this? So we need to understand why CAR-Ts stop working, because unfortunately there are people who have progressed after CAR-Ts. We don't know how long it's going to last. Obviously we're still sort of in the early stages. Having said that, I do have a patient that just came in for her 2-year follow-up and is in remission after having her CAR-Ts. So we know it can be durable. So again, understanding who responds best, who doesn't. There's going to be next generation CAR-Ts already happening. So there are some CAR-Ts that are looking into making your T cells more like memory T cells. Is it important that maybe these CAR-Ts, we don't know how long they last in your body. Do they need to stay forever? Do they not? So some companies are working on CAR-Ts that have a phenotype more like a memory T cell that won't fight your disease right then and there, but may sit in your body and come up only if your disease tries to come back. Safety is a big issue, right? So how do we make this more accessible? Is it something that can only be given in a transplant center, for example? Is it something that's going to be available to you further out? That all depends on how safe it is. And so trying to figure out which patients have CRS, which patients get neurotoxicity is very important, and that work is still ongoing. We still don't know who is at risk, although there's some ideas and some early data. But there's actually new CAR-Ts that have safety switches, where you can actually infuse a CAR-T, and if it's really dangerous, you can actually give a drug that actually kills the CAR-T, and so you can turn it off. And so those are coming, so hopefully that will make these also a better or a safer product. And then improving the access, of course. Right now you have to collect your own T cells and make them into CAR-Ts. Would it be nice if you can just actually buy the drug just like you do with your Darzalex, where you get allogeneic CAR-Ts that once you're ready, you don't have to wait that month for the CAR-Ts to be made before they get infused. You actually can just be given the drug as soon as you're ready to give it. And so these off-the-shelf CAR-Ts are very early, but they're starting. And then actually moving the CAR-Ts further in. So just like everything else, we start in the relapse refractory setting, but as we kind of get better with it? Do we really need to wait till you've exhausted all your therapies? There are trials now starting to open in sort of the relapse setting, and even in high-risk patients, perhaps in the frontline setting. So again, the CAR Ts are moving forward as well, just like everything else in myeloma. Key points, I mean, obviously everyone's excited about CAR Ts, but remember there's still a lot we don't know about it. They can be very toxic, there's limited availability, but I think it's moving very quickly, so hopefully it will become much more available to all of us. We don't know the long-term outcome yet, so you know, it would be nice if we knew that it works and it's going to work for forever or for 5 years. If we can get, you know, a break of therapy 5 years, I think we probably wouldn't mind, right? Anyways, we still don't know how long that will happen or will last. There's different potential targets. There are some patients that seem to progress and they lose the BCMA, so do we need to go through after other proteins? And then, of course, the antibody-drug conjugates I talked about earlier also are very exciting. So I think I have 2 questions before I stop. So this is just an easy one, true and false. So there are no FDA-approved immunotherapies for the treatment of multiple myeloma, true or false? False, okay. Yes, false, right? So we have Darzalex and we have Implicity. Those are immunotherapies and they're FDA-approved. So most correct, that's good. Which of the following are types of immunotherapy being developed in multiple myeloma? Therapeutic vaccines, antibody-drug conjugates, CAR Ts, bispecific antibodies, monoclonal antibodies, or all of the above? Good. So yeah, so all of the above. Excellent. You are all paying attention. I'm impressed. All right, I'm done.
Oh, you're welcome. There are really a gazillion questions here about immunotherapy, mostly about CAR T cells, and a lot of them are about the timing of CAR T cell therapy and questions like where is it going to go? Is it going to go into the front line? What's the best places to do this kind of stuff? Mostly questions that are not answerable questions, but you could expound on some of more detailed about where you think it's going.
Yeah, no, so I mean, I alluded to this in the summary slide a little bit, but I mean, definitely in the relapse refractory setting, that's where it's starting, but just like everything else, there are clinical trials coming down the pike that are moving this earlier in the disease. So there is going to be a randomized trial that's going to open, believe it or not, in the 2nd or 3rd relapse. There's actually potentially a trial that's going to include patients with sort of what we consider super high-risk disease, even in first remission. And so there's also going to be combination trials. We don't know, can we actually, you know, help again, your immune system boost a little bit further, so combinations with CAR Ts and others will be interesting. And in lymphoma, actually, where they're a little bit ahead of us, they have a trial with CAR Ts and one of these checkpoint inhibitors, again, trying to overcome that potential resistance that may develop to the CAR Ts. So no, the answer is, I think they're going to be in all aspects of your disease, from the newly diagnosed to relapse refractory, but it's going to be all in clinical trials. So again, important that you know, David have tons of clinical trials for you. They will actually have a lot of these as well, and other doctors in this area, of course. You're all very lucky that you have all these centers because they will have these trials for you. Which is not the case always in other parts of the country.
You know, one of the things that I would say is that we see treatment decisions changing and patients' ideas about what they want to have done based on the anticipation of CAR T cells. I think that it has to be understood that we still don't know the long-term outcome from CAR T cell studies in myeloma. I mean, we know that it works well in the short term. We don't know what the long-term results are. And more importantly, the big trial in the U.S. right now, called the KARMA trial, is going to have somewhere north of 100 patients. But it's not just in the U.S., it's around the world. And again, it's 150 patients. There are 100,000 patients with myeloma in the United States. And the idea that that it is going to be routinely available in the short term is not the reality of it. So when I see questions here like, when should I get my CAR T-cell therapy? Well, I wish that was an answerable question. It is not. The person who's done the most of these bluebird type transplants, the original bluebird study, and now the karma study that has extended out of that is Jesus. And how many patients have you transplanted on those two studies?
Yeah, 15.
Yeah, so understand that this is not the answer, partly because it's not available. So these kinds of questions that are there are not questions that will be answered anytime in the near future. And which CAR T-cell therapy is better, which vaccine therapy is better, all of these questions, we don't know the answer. The vaccine therapy, we have no idea whether these work at all. There's some hints that we can change the immune system a little bit. Whether that translates into long-term benefits or important benefits, we don't know. This is stuff that we're just starting to learn. Anything here that you see in this enormous list of questions that you think is something to expound upon? So, as I said, these, do I do it in remission or relapse? You do it where the clinical trial will allow you to do it, and that's very, very limited right now.
Well, one question is, can blinatumomab be used for myeloma treatment? The answer is no, although in theory there are CAR Ts directed against CD19, which is what blinatumomab is directed against, that we know have some activity in myeloma, but truly the vast number of myeloma cells do not express CD19, which is what blinatumomab goes after. So no, the answer is you can't just use it as standard of care in myeloma.
And most of the availability of these things is based on geographic location. Why does Hackensack have this trial and not not Valley Hospital? Well, because I know the people who talk about the trial, and they don't. So there's no sense to any of this stuff about why and how these things become available.